I don't. And you see me? I just can't. I go to be like the same person. I know you're not seeing me in home five. I just can't ball. I can't do it. My games and my whole crisis. I want to do them. Wow. Wow. I know a little too much. A little too much. I don't trust this school or minutes to like fighters. So, oh, you could do it again. Yeah, stay my balls, right? So, what you tell people not so? So, like, you could that one, right? You heard the shit with someone? So, you could have that one, right? So,
There's so many dates and time on, I guess.
right. I can't tell you. So, I don't tell you. So, the first, so September, so just search for that September, then you will find it.
And you sit on the bangers. Me when he was going through it.
So you want me to get you to him?
No.
So who would have you do?
But he goes to tell them on pass, dog, on the pass. He said, well, you can't tell them on the pass. Well, why you asking who? He clearly says, yeah.
Huh? Yeah.
You're not using very beautiful language, huh? You're not using very beautiful language.
You guys said you already know. I watch it. I'm trying to figure out what I did wrong.
I did this good things too.
Only thing I did wrong was
Brother, we go for graduation. They say there's good things. They said there's good things. Hear what she said. Brother,
So remember we had finished with module 1, right?
wait,
so we-
graduation too.
We started with module 2 now. Right, so module 2, I'm plenty to do with the genes and genetics. Can we all go ahead now, right? Okay, genetics. I can't even make a joke if Shakira actually tell me what happened. And he got
No, but you can still do.
I prefer not to. Right, so we're starting with the genetics.
Chan, my graduation. Oh, wow.
Okay, so we start with some revision, right? Your genetics or genes in all living organisms are made up of something called nucleic acids. Right? And there are two types of nucleic acids. Right? You have DNA and you have RNA. Right? DNA stands for deoxy ribonucleic acid, and RNA stands for ribonucleic acid. Right? Both of them similar in structure, but the one difference they have plays a big. Yeah. Right. So your DNA is found in your genes, and your RNA, think of it as like a messenger or an intermediate between your DNA and proteins. How your genetics work, you have stored DNA. Your DNA is like information, and that information is used to make proteins. Right? Now, based on the proteins that you produce, they'll give you the characteristics. So it could be physical characteristics like height, things like that, right? It could also be characteristics like blood type. It could be characteristics like certain diseases. Right? All of that is inherited through DNA. Your RNA is RNA is used to produce the proteins from the DNA. Think of this as like a template. RNA translates the template and makes proteins. Well, helps to make proteins. So we know the structure of these nucleic acids.
So we don't pay for your window, just we pay for the computer. I know. That's the initial. What you're doing, you had such a restaurant, just.
Yep. Yeah, but as a she on just message. Tell her record it on. Something.
Yeah, share valuation.
Yeah.
Cannot.
You supposed to know. No.
Oh, let's not talk about this.
Okay.
Because I come very
So this is the structure for nucleic acid, right? So this is what you call a pentose sugar. Again, pentose sugar can have 5 carbons. Pentose sugar. A pentose sugar is attached to a nitrogenous base. This simply means it have nitrogen in this structure. That's all you'll need to know, right? Have nitrogen present here. And it's also attached to a phosphate group. This is the general structure of any nucleic acid. Have pentose sugar, nitrogenous base, and a phosphate group. Okay? Both of them have the same general structure. Right? The difference. The difference in structure between deoxyribonucleic acid and ribonucleic acid is in the pentose sugar itself. So if you look in the name, you'll see DNA deoxyribose. This means deoxy, it lacks 1 oxygen. So it has 1 oxygen less than ribonucleic acid. And that's the difference between the two in structure. So it will be found on the pentose sugar. And these strands are not to see, right? So, tell me agree.
Yeah, she teaches a lot.
I don't know yet. It depends on all of you.
Exactly. So what emotion? Oh, yeah, that's the old.
So, brother, you scared what I
am
do?
scared, dude.
I was in my name in it, but nothing happened. I could see.
About the situation.
Nobody said anything wrong, Joe.
It's very beautiful.
So I met my mother earlier.
Huh? We messages. In the office? But she know what's a beautiful sunshine.
I'm confused.
Legit.
Okay, right. So watch up here, right? Hey, it's your watch up. Right. So this is the structure for DNA. right? This is the pentose sugar for DNA, yeah, and this is for RNA, right? You'll notice that on the second carbon, right, which is here, ribonucleic acid has the OH bond, right, or OH group. DNA lacks that OH group, it just has a hydrogen, right? That's what gets the name deoxyribonucleic acid. Right, so that one little difference changes the entire structure or entire function. Right, so take down these structures for both DNA and RNA. Again, it'll have a phosphate attached up here and a nitrogenous base attached as well. Right, so these are individual nucleic acids.
Where is it? I am checking it. Push him out.
Is an O here just so the O is at the top here. Look, above. Obviously.
Oh, yeah, you didn't teach them. Yeah, so you said, oh, well, this.
So he's the example teacher?
When you're.
They won't see the sound.
I didn't know what they
I
say.
say that I don't join that. Wow.
Oh, stay up.
He know me, I know, I'm used to that, no? I give him this room.
When we did, you put C all over it, and if it's not put it, okay, that equation. And you put C.
Yeah, remember at each edge is a carbon, right? I just didn't draw it too.
I was here. Oh, yeah, your phone.
Just make a change to the drawing, right? Put the key on this sugar, on this carbon, sorry. So move from here to here. He would be on this carbon. Right, so these are individual nucleic acids. when they join together, when the phosphate group from one nucleic acid joins to another or forms a bond to another, they come together to form a polynucleotide, right? This structure here is called a polynucleotide. And that's when you have multiple nucleic acids that are joined together by phosphodiester bonds, right? Or phosphodiester, phosphodiester. Right? So it attaches phosphates to the carbon, or latter phosphodiester. The nucleic acids are polynucleotides are held together by phosphodiester bonds.
Actually, I saw them some persons.
She says I'm a soldier. I sold I sold somebody. And I tell him he can't do it.
No, you don't.
What? Tell him I sold somebody. That don't sound like me, you know. That should not sound like me. I never tell it. If I know you're assaulting somebody, I'll easily drop it. That don't sound like me. That don't sound like me. I don't know why you stop. So a person is a person he assaulted. And you beat me up for that.
I'm not involved with anything.
Is that girl though?
Why would it be a man?
It's Jordan.
Is somebody in we class?
Nah,
We assault? Is somebody in we class we assault?
Hey, this being recorded. I'm just saying.
Yeah, it was important.
No, I'm
So who are
saying
so who are so
what?
who are so
Legally, I can't say
Yeah.
anything.
But he was like an apocalypse.
All
All right, so I'm sending you all a handout, right? You all will check it out.
Oh, my life and my name can
Yeah, yeah,
this is a lie.
It never that deep.
Sir, when I go to the street, like there's you,
Camille, you don't go on the street. I don't know what you're trying to. You don't go on the street.
Jordan, I saw that I see.
Sir, I can tell by passing consent.
It's
Hmm?
Chaya.
I'm calling now. I'm calling.
It's Chaya. It's Chaya. You assaulted.
No.
Camille,
Is she
Be quiet, please.
Like you.
Camille.
It's the only.
Yeah, so take it.
All right, so you all look at the handout, right? The handout is on nucleic acids and the genetic code, right? So I set it on WhatsApp.
But I wouldn't do them things. But I wouldn't do that. I would never do something like that. No, but this one's serious. If it's something somebody knows. My mother raised me right, you know.
All right, so let's just take a note on that now for the structure of nucleic acids. So Yes, all right, sir. So again, these are like shortened. There's a piece of chicken you take one. Huh? Oh. One, two. Right. So again, remember, the notes that I give you all are like concise versions of what we would have in textbooks. So no, even the thing essential. So use the two of them. Don't just use one. So write in DNA and RNA. DNA and RNA. And nucleic acids. And nucleic acids. Which are comprised of a pentose sugar, which are comprised of a pentose sugar, comma, comprised of a pentose sugar, comma, a phosphate group, and a nitrogenous base. A phosphate group and a nitrogenous base. Also. Individual nucleic acids. Individual nucleic acids are called nucleotides. Individual nucleic acids are called nucleotides. And when they form phosphodiester bonds, between each other,
Excuse me, you can smell phosphorus.
pH. No, wait a minute. Tell me what. Yes, you know, the young and the isotope be strong.
Yeah, but it's a
Or should you need to say one? Right? And so when they form phosphodiester bonds between each other, they form polynucleotides. Full stop. And skip on a line. The next sentence is the structure of DNA. Structure of DNA. Right, so we're going to see that real structure of DNA, right? So now, the nitrogenous base part of DNA. So this part could be one of four groups, right? Those groups could be adenine, guanine, cytosine, or thymine, right? So you'll have the sugar base that's attached to either adenine, guanine, cytosine, what time?
Very small. No, very small.
Okay. So this is adenine. Okay, right? So again, DNA could only have 4 bases attached to the individual nucleotides. Rather be adenine, guanine, cytosine, or thymine. Right? You would also be familiar with seeing this structure of DNA at some point in a movie or something you would have seen. No, like, who say no there? Yep, 400.
Just like the thing on the stairs.
You are trying.
You got it. Right. Good job. Close.
Right, you're accustomed to seeing something resembling
I'll best of like.
it, right? So the reason for this is DNAs have a double helix structure, right? Double helix meaning it have 2 strands that are running alongside each other, right? In between these strands, you have the bases. So the line that you see here is the sugars, right? The sugar backbone, and in the sugar backbone, you have your nitrogenous bases. Right, so for example, right, this is your sugar backbone, the pentose sugar, and attached to that pentose sugar would be a certain base. Let's say that base is adenine, right? These bases are complementary to each other. So the complementary bases are A is complementary to T, and G is complementary to C, and vice versa, in any direction. So if in DNA you have adenine on this side, on the opposite side of the double helix, you will have T. Okay, they are complementary to each other, they communicate. So if I have G here, which base would be complementary to C? C. Right, and if I have T here, which base complementary to E? You understand? The double helix is 2 separate strands running alongside each other. Another thing we have to be familiar with is direction. Okay, so how do we describe direction? No, we're not do that yet. So take down what we have on the board so far. So again, this is structure of DNA.
This is BBC watching on the web, right?
Yeah, that's for RNA. So thymine is found in DNA, but it is not found in RNA. And use for uracil is found in RNA, but not DNA. Yep.
And this hold on, this TAC.
Yeah.
Because I was looking up question and I was trying to figure out where Google was being something.
No, use TAC. Well, for which one? For RNA?
I'm artist, which I could go be to see them. What's for the other?
No, what I mean is, remember we had the start codon would be on RNA, like the mRNA, and then you'd have the anticodon on the tRNA.
Yeah.
So one of them is TAC, it's one RNA. I think the TAC one is on the mRNA, and the other one the U. So it would be on side.
But it's going to be on glass here.
Yes. Think so. Right, so just take down this. These are the complementary bases in DNA. These are the 4 possible bases in DNA, and it's the double helix structure of DNA.
Don't like me though. Don't like me so. Just be the classroom.
Camille, do you want?
So just plug me over. So I have a message launch for this.
What's up, if you're like, just feel
Something.
Actually, go home and have to coach up.
That's
That's scary. Just somebody too.
Wait, calling your mommy.
You should be cool. Nada, we put on site to.
Yeah. Please, let's get out here. The not me over here. That's getting
Some wrong.
so, cause I didn't do something wrong, but Kira said disappoint she, but what did I disappoint her and put me to do?
Search within yourself. You'll find the answer.
Legit down.
It's context, not even speak.
Did not.
So even public school, they go and take water, so I can't national
No worries. I'll
So
say it.
no, so if I actually do something, I go and get kick off, so I can't do nationals again.
Nah, I'm just
No, my mom would take water too. No, she wouldn't.
we already
How
told
you
you.
doing for that? So
And if you're not doing it, what to you take you out?
Because I don't know what's happening. I go back.
Right, so while on the drawing that, you'll see on the handout, it has some features of DNA, right? The first one is double stranded. Because of that double stranded nature, it allows it to be a double helix. Okay? Now the double helix between the bases, they form hydrogen bonds. Right? Jordan, you're not getting in trouble, man. Just stay quiet. Huh?
I don't even trust you anymore.
Just stay quiet. Right? So between the bases, so like for the first one, A and T, they form hydrogen bonds. That's what keeps them together. Right? And again, they're complementary. Those are the four main features of DNA. Right? Double stranded, double helix, form hydrogen bonds between bases, and they have complementary base pairs.
So I was meaning to ask you this, but I couldn't find it in like in a past paper.
Mhm.
They asked you to draw the deoxyribose DNA structure, the ribo DNA structure, and them thing or
They could, but most of the time they'd ask you to describe it.
just describe it. Okay.
But you should, once you could draw it, you should be able to describe it. Right?
Yeah, but I don't need to know the answer to the
Or it's a zip thing.
yeah, I was going to ask you to describe it.
I like it.
Make it so, it's a whole reason it's like to see it.
I understand it
Yep.
From now on, and make sure I give no show break now. I start even just again.
But you give it trouble, all know,
Yeah,
So-
you know what problem I got?
what problem?
And I saw my gear on assault.
I should see your
I
photo.
saw somebody.
No, she meant you harassed her. That's what she meant.
Something like those.
My head.
Forceful.
You're saying, you're in the group. I'm on the phone.
It's from Kiran, all this kind of I don't feel like the most respectful person.
But if you respect me.
Yeah, I don't respect
Nice
it.
here.
You are you. Yeah, Kira.
Oh, you.
But I'm here.
I don't create it right now. I'm talking to another one. I don't get you. You are in a bar for just to I was in the
Because you know, almost like to be like.
So you have a seat, sit in front.
Look, we see no.
Yeah, that's you. That's no. I didn't do that. I just didn't do that.
So you can't give me that, please.
Hold on.
I want to. Oh, so here it's all about.
You see how bad we are. It's not as serious.
It's short.
Right? Don't worry.
I just want to see that.
I don't know you. Well, the chat, there's that chat going.
Wait, the man just see it, not having something. Why not coming with big one, big one? So,
Hey,
I'm sure it's.
Jordan, let's focus on your for now.
I focus on how we. Well, we got that something, somebody says.
Hey,
You see, so it's that.
you control. Jordan, if you can't focus, stop disturbing the ones around.
这那个我在一起的时候，
还没有算对了，他不合格啊。
Nem uma bicocha, né? Já vou ter que
Ah.
demais.
Alright, so watch up here, right? So this is just the phosphate, the sugar backbone, right? And these are the bases inside. So this is like if you zoom in, the part of the DNA, right? Okay. And I want you to look inside. So you'll see in the complementary base pairing, right? These bases are held together by hydrogen bonds in pairs. Okay. Now on some of them you'd see 2 bonds or 2 lines, and some of them you'd see 3. Between cytosine and guanine, it forms a triple bond. So that's why it has 3 lines. Between adenine and thymine, it forms a double bond. That's why it has 2 lines. Okay. Another thing to note too is direction. So this will be going in 3' to 5' direction. And this one will be going in a 5' to 3' direction. Yeah. How do we get direction? What you're looking for is for the carbon, which is closest to the end. So for example, this is your sugar compound, right? Or your sugar backbone. The first carbon would be where? Right, so this would be 1, 2, 3, 2. Oh no, this direction only. This will be 5', 3'. And this will be 3' to 5'.
Yes, it was
Yep, right? So you're looking for the last carbon here. So you start with the first carbon, 1, 2, 3, 4, and the 5th one will be somewhere here, right? Remember, the 5th carbon is off the 4th carbon. So the 5th carbon is closest to the end. So that's how we get the 5'. You understand? If you label the carbons, you'd see it has CH2OH on the side. So the carbon closest to the end is the 5th one. That's how we get the 5'. Now, if you look at this side, we started carbon 1, carbon 2, carbon 3. So carbon 3 is the one closest to the end. You want to see that?
Stop.
Right. So remember, this is the structure of right? So this is it with the actually label, right? Where do you start counting in a pentose sugar? Which one is with the first carbon? Start on the right side. So this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Right? Now let's imagine that this is the carbons labeled here. Which carbon is closest to the end of the chain? In this one, which carbon is closest to the end? The 5th one, right? So that's why we call this 5'. Now if you come over to this one, at the other end of the chain, the 3rd carbon is closest to the end. So we call that the 3'. Okay? Now if you go to the other side now, this is the other strap. You'd see that the sugar phosphate backbone, or sorry, sugar backbone is split. So in this carbon, right, where we started to come from? We started to come from this side, right? The side opposite the phosphate. So see the phosphate here, we started to count here. We started to come in the side opposite the phosphate. Okay, so this will be carbon 1, carbon 2, carbon 3. So this is the 3'. Okay, now if you come down, again, where we started to come from, the carbon opposite the phosphate. So it'll be 1, 2, 3, 4, and the fifth one somewhere. So this will be the 5 prime. Right? So then we call this an anti-parallel strand because this strand going in an upward direction, this strand running downwards. So it's anti-parallel. Okay? We all understand how to get direction.
Yes, sir.
So all you went through structures so far. Sugar phosphate backbone, attached to a phosphate and a nitrogenous piece. That's the structure of any nucleic acid. Right? After quality, so we always go back again. That's how it is. Right, so take down this as well with the direction. Right, and after you finish with that, you can just put the heading structure of DNA. Right? Have a little note. You do something else, right? You know, this so. I mean,
Oh, you make it.
This one hard, I'll tell you, this one, more fun.
yeah, I like that people. They are grateful. With Arshad? I'm grateful. Is this good?
Listen, I tell you, it's not my first beer on that.
Huh? Already bites it.
You guys break up, yeah.
I know you have enough.
Blew up.
I know you have enough hard day. Show it to me.
So I know it was involved.
Hey, Jordan. Have you come there and class, right? You have to class if you have an actual person. Okay, Jordan.
You're getting it from the company too.
Yeah.
I mean, this is good.
Like,
Yeah, you go.
okay, you
Let me find it.
really go see that old shit. Those such things.
Hey, I don't know the right time. Like, I think you missed something. I look like this, was no way to do something. Without be shit.
No matter. Once the right thing's connected to the right parts. Okay. Right, so we take any note for it, right? The structure of DNA. The types of bases, the types of bases, the types of bases, types of bases that is attached to the sugar backbone. The types of bases that is attached to the sugar backbone in DNA are adenine, comma guanine, in brackets, purines, so that adenine and guanine are what you call purines, right? All that means is they're slightly larger. Adenine, comma guanine, and in brackets put purines, and close brackets, comma cytosine and thymine. Cytosine and thymine in brackets, pyrimidines. Right, so they're slightly larger than the pyrimidines are slightly, the pyrimidines are slightly smaller than the purines. So thymine and cytosine would generally be smaller than guanine and adenine. Right, full stop. These bases are complementary. These bases are complementary. These bases are complementary to each other. These bases are complementary to each other. And held together by hydrogen bonds. And held together by hydrogen bonds. Full stop. Next paragraph. Two DNA strands are held together. Two DNA strands are held together in a double helix. Two DNA strands are held together in a double helix. Which help to make the DNA molecule. Which help to make the DNA molecule very stable. Which will help to make the DNA molecule very stable. Full stop. This is important. This is important. as if DNA changes. This is important because if DNA changes, comma, this can lead to serious illnesses. This can lead to serious illnesses. Full stop. Right, so once the DNA that you're born with, the genetic information you're born with, you'd have it for the rest of your lifetime. If for some reason there's a change, let's say a mutation, most mutations are deadly, right, or dangerous. Right, so if you have a change in your genetic makeup or change in these bases, that could lead to a change in the proteins expressed, which could lead to a disease. An example of that is cystic fibrosis. Any of you will ever hear about cystic fibrosis? Yep, right. So it's basically when your lungs filling up with mucus, right? Is a genetic disease, which means a mutation happened somewhere along the DNA strand, and that caused too much mucus to be produced in the lungs. Now you don't want that because you need the lungs to expand to breathe, right? To get oxygen in. If it filled with mucus, you could drown, basically. Right? So that'll be an example of a genetic disease. We'll do that later. Stop. DNA strands are anti-parallel. DNA strands are anti-parallel to each other. Which means they run in opposite directions. Which means they run in opposite directions. Full stop. The 5' end. The 5' end is the end where the carbon 5, the 5' end, is the end where the carbon 5, of the pentose sugar, is nearest to the end, while the 3' end is found where the carbon 3 of the pentose sugar, is nearest to the end. Full stop. Any questions on the structure of DNA? Right, so, yeah.
So like in the green DNA strand, right? The term goes to the 2' to 5', so I want to know what's what it.
You mean if they don't give you like anything? Well, they'd have to give you at least one of them, right? So if they just give you this strand, let's say they give you this strand, right? If they label this 5', you're supposed to automatically know, okay, if they start with a 5' in there, this has to be 3'.
I'm saying like, here I can. Horizontal, I don't know what I'm right.
Like this?
Yeah.
That is up to you. Right, so if you want to start this as the five prime, then it's arbitrary.
What's your amount of that?
This one will be. Three prime, five prime. This one will be the second one. Right? So it'll be five prime to three prime to five prime. It don't matter how you write it. What matters is that it makes sense. Okay? As in, if you have five prime here, the end of that strand have to be three prime. And the strand opposite it have to go in the anti-parallel direction. Yeah? Understand what you're trying to ask? How would this relate to that? Right? But there's a horizontal drop as a vertical. Yeah. Right? So it's two different things. Right? But they would not ask you to relate the two of them. Right? It's impractical. Right, so why is the DNA found in a double helix? What's the significance of that? What about the double helix is so important, or why is the double helix important for the structure of DNA? The stabilization, right? You do not want your genes to change or your DNA to change. So because it's in a double helix structure, it's like protected. The base is not exposed. Right? And where would you find DNA? Which part of the cell? The nucleus, specifically, which part of the nucleus? The nucleolus, right? Right. So now we're going on to the structure of RNA, right? RNA. Now, RNA is a little bit different from DNA because it doesn't have the thymine base, right? Instead of thymine, it has uracil, represented by U. Okay, so it still has the pentose sugar, but this time it's ribonucleic, right? It's ribose sugar that is attached to these four bases. Okay, and instead of being in a double helix, RNA are single strands. Now they still run with a 3' and 5' end, right? It still have the pentose sugars, but it exists as a single strand. Right, big difference is that uracil. Right, so with DNA, it's just DNA, right? It have one type. The DNA is just a DNA. So RNA is a little bit different. There are three types of RNA that we use: messenger RNA or mRNA, transfer RNA or tRNA and ribosomal RNA or common RNA. Right? We have to go through each of the different types. Well, except for ribosomal, right? That one really on the cell, well, not on the cellulose. Okay, so right, so there's just a simplified drawing of a transfer RNA or transfer RNAs, right? Now, this is not a double strand, this is a single strand that folds, right? There's one single strand that just folds alongside the strand. They don't have more than one strand in. Now, the important part of a transfer RNA, right? It has two big parts. At the top of transfer RNA, you have an amino acid that attaches to it. Okay? The amino acid that attaches here is based on the anticodon that's down here. When we do protein synthesis, we'll understand what that means. But for now, let's be familiar with the structure of tRNA. The amino acid attaches up here, and this is like, think of this as like a barcode scanner, right? The anticodons would have to read the codon. Once they have a match, then it could donate its amino acid to. Okay? We'll get into what I mean. You all need to know what that is as yet. Yeah. Right? What do you mean? This side attaches to the mRNA. This side is just attached to the amino acid. Once it have a perfect match, then it can give up its amino acid. Okay? Okay, so for now, let's take down what I have on the board, right? The three different types of RNA, as well as this diagram of tRNA. In the handout as well, it would have the structure. Yeah, I don't think the drawing, but it's on the handout. Yeah. Just try back and that's simple.
That's what it's trying.
Right? Right, so the side with the amino acid with the 3' side, and the side with the 5' side. Right? When I'm doing it back in protein synthesis, I'll put it. Because
Yeah.
like when you all did it the first time, I realized you're all confused. Because you all didn't know what was the signal. So I'll come back. Right, so in the RNA strand, in tRNA, you would have base pairs, right? Within the strand itself. So if you have A here, you would have a U here. If you have G here, you would have C here. If you have T here, you would have A here. Now, I do something wrong in this. You can tell me what I did wrong. T, right? This is not supposed to have a T. Why can't this have a T here? So this is RNA, right? So it's not supposed to have like time. Supposed to have years. Right? So again, the complementary base pairs would form hydrogen bonds between each other, just to hold together the tRNA. But again, it's one single strand that's forming the tRNA. Well, because it folded against each other just. So for example, if you have like this strand, right? This one single strand could fold. And remember, along these strands, it have bases. So if it folds, if one strand folds at some point, the bases going to interact with each other. Right? And the complementary bases in that fold would pair. Okay, so after you finish taking on the drawings, you can just put the structure of RNA, right? The structure of RNA.
Yeah. Right, okay, to the ball, I'll take that, so.
Okay, right, so again, structure of DNA, so you're writing, sorry, structure of RNA. RNA are made. RNA is made it's made from the four bases of adenine, comma, guanine, comma, cytosine, and uracil. So adenine, guanine, cytosine, and uracil. Full stop. They form single polynucleotide chains. They form single polynucleotide chains. And tend to be shorter than DNA. Tend to be shorter than DNA. Full stop. There are three types of RNA. Colon, right? Messenger RNA, transfer RNA, and ribosomal RNA. There are three types of RNA, colon, mRNA, tRNA, rRNA. Full stop. mRNA is formed in the nucleus. mRNA is formed in the nucleus comma, is involved in protein synthesis, comma, is involved in protein synthesis. And has a very short lifespan. Full stop. tRNA has a single chain. tRNA has a single chain which is folded into a clover leaf shape, CLOVER, a clover leaf shape. Full stop. There are different types of tRNA. There are different types of tRNA. There are different types of tRNA. Stop. There are different types of tRNA. The main structure is always similar. The main structure is always similar. Except for the main structure is always similar, except for the three bases, except for the three bases of the anticodon, except for the three bases of the anticodon. Which determine the amino acid, which determines the amino acid that is attached. Determines the amino acid that is attached. Full stop. It is also involved in protein synthesis. Is also involved in protein synthesis. Full stop. Also involved in protein synthesis. Full stop. rRNA is made in the nucleolus. rRNA is made in the nucleolus. Full stop. Right, so the ribosomal RNA, you all don't need to know about, you just need to know the thing. You all don't need to know its function because it doesn't really play as big of a role in protein synthesis. Right, so what I want to do before we move on, is just compare RNA to DNA. Right, so just doing a table to compare the two. Okay, so the polynucleotide chain of DNA, right? Is it a double or single chain? Right, so in DNA, have a double polynucleotide chain. What about RNA? Single. Right, so we have to find different ways to compare it. What about the lifespan, right? So what kind of lifespan would DNA have? Relatively long, right? So as long as the organism is alive, it would continue DNA. Same unchanged DNA. But that's a long, or you put lifetime, right? So however long that organism lives, it should continue same DNA. What about RNA? Right, so it'll be shorter. Depending on the type of RNA, it'll determine how long the lifespan is. But regardless, it's not going to be short. Right, the next comparison is the bases. What bases do we find in DNA? Right, so A, T, G, C, and R. A, U, G, C. Right, so those are the bases. Right, what's the next difference? So I just listed differences between the two so far, right? What's the next difference between DNA and RNA? So what you want to come up with the check out?
How one and how-
Correct. So the types, RNA is just one. So DNA is just one, RNA is three. What's the next way you can compare it?
yeah, it's constant, like species, but it's very.
Right, so DNA is constant within the species, within a certain species, and RNA is variable within the same species. Right, so what does that mean? It means, okay, if I have a skin cell, the DNA I find in that skin cell would be the same DNA I find in like a nerve cell. Okay, now if you compare RNA, right, the RNA in my skin cell would be different from the RNA that I find in a nerve cell. So within the same species, the same organism, the DNA would be the same in each cell, and the RNA would be variable, depending on what the cell needs. Right, so obviously your skin cells would be involved in protection. Right, so the RNA that's being expressed or used to express the protein would help with protection. The nerve cells, their main function is what? What the main function in nerves? Transmitting signals, right? So the proteins are expressed in nerve cells to help with electrical transmission or signal transduction. Add an example.
No, right, so.
Right, so the pentose sugar, right? Pentose sugar, right? So this would be deoxyribose. I just write in deoxy, right? You're right on both, actually. Now deoxyribose sugar in DNA, and you have a ribose in RNA. So these are the main differences between DNA and RNA. Right? What about direction? Is direction a difference between the two? No, I mean like, right? Both of them have the same direction. So that's what I mean, right? Both of them have a 3' end and both of them have a 5'. So it's not a difference. That's what I'm trying to tell you. It's a similarity. So you only need to know similarities and differences between DNA and RNA. So these are different, right? Yeah, just look at it, right? Right, so I don't want to start anything too extraneous. Because the class nearly done, but we could just focus on this for now, right? So this is to do with the structure of DNA, right? So we have to know the different, well, we have to know how to describe not just the structure of the DNA, but also of a chromosome. Okay? So yeah, I'll zoom. Okay. Yeah. Right, so this is your chromosome here, right? This is your chromosome. right? And in the chromosome, if you zoom in on a chromosome, you would see the actual structure of it, or what it's made up of. Right? So again, this is along your chromosome, and along your chromosome, you'd have DNA molecules. These DNA molecules, scroll up, right? These DNA molecules form histones, right? So you see these little green things wrapping around them. When you have DNA molecules wrapped up, we call those histones. Okay? Right. So one of the structures is called a nucleosome. Down. Right? So one of the structures is called a nucleosome, and it's just DNA molecules that is wrapped up. Right? And many of these, when they coil together, they keep on coiling against each other, they would form your chromosome. Okay, so I'll send this picture to you all. This is the structure of a chromosome. Again, it's made up of DNA and histones. Histones are really proteins, right? So the green thing is just proteins that wrap around the DNA. So I'll send the picture to you all so you'll have a reference. And we just take the note for it now, right? The short note. Right? So the heading would be structure of DNA and chromosome. Yeah. Just end with structure of DNA and chromosome. So anytime they ask for structure, just think of this diagram. Structure of DNA and chromosomes. Okay, so writing chromosomes are made up of chromosomes are made up of chromosomes are made up of DNA. Chromosomes are made up of DNA as well as proteins. DNA as well as proteins. Full stop. The proteins that are found in chromosomes, the proteins that are found in chromosomes are called the proteins that are found in chromosomes are called histones. The proteins that are found in chromosomes are called histones. Full stop. Called histones, full stop. The histones group together. The histones group together to form a nucleosome. The histones group together to form a nucleosome. Full stop. Each nucleosome contains 8 histone molecules. Each nucleosome contains 8 histone molecules. Wrapped around DNA. 8 histone molecules wrapped around DNA. Full stop. the nucleosome eventually loops. The nucleosome eventually loops and coils. The nucleosome eventually loops and coils to form a solenoid. Form a solenoid. Which further loops and coils. Which further loops and coils to form further loops and coils to form chromatin fibers. Which further loops and coils to form chromatin fibers. Which further loops and coils. Which further loops and coils to form chromatin. Which eventually supercoils. Which eventually supercoils. Eventually supercoils and fold. Which eventually supercoils and fold to form a chromosome. Which eventually supercoils and folds to form a chromosome. Now, I make a mistake just now, right? The DNA wraps around the histones, not the other way around. So the green part was the DNA, and the blue part was the histones. So the DNA is the strands that wrapping around the histones. But the two of them together is called a nucleosome. Right? So after some coiling, a good bit of coiling and folding, it ends up from a nucleosome to a chromosome. Okay. Any questions from today? Anybody? So that's it from today. Kemuel and Jordan, just stay back for like 5 minutes.
Yes, sir, that's it. I'll check the show.
Okay. All right. The rest of y'all, that's it for today. We'll continue next week. What I want you all to do too, if you all get time, to just read through the part with DNA replication and protein synthesis in the hand, just so you all could have an idea of what it is for next week.